Hello, and thank you all for attending the third and final installment of the InnovATEBIO CRISPR webinar series. A couple of housekeeping items for you all. We do have live transcript available for this session. You can access that by clicking on the live transcript button at the bottom of your Zoom screen and selecting Enable Subtitles. Please feel free to use the chat box to ask questions. And the presenters have also indicated that they would be open to taking questions directly during their presentations today. We ask that you do keep your mic muted if you are not speaking. And with that, I will hand it over to the excellent set of presenters that we have today.
Well, very good, Heather, and thank you very much. My name is Kmiec, and I'm the director of the Gene Editing Institute at Christiana Care, which is a large health system that located in northern Delaware, serving Maryland, New Jersey, southern Pennsylvania, and Delaware. And it's a real pleasure for me to be participating in this wonderful program. I'm going to be joined today by my good colleague, John McDowell from Delaware Technical Community College. And my role is essentially to give you a bit of a 50,000-foot view of how we develop CRISPR in a box, and also to talk about how some of the new things that we're going to be launching today might help you with some more teachable moments around the technology that you can use in laboratory. So if I can have the first slide up, I just want to kind of give you a little bit of background into our system. We are the only institute of our kind in a community health system, although I know that's changing fairly rapidly. We're 5 years old. And once we started this, it became apparent, I think, to people in health systems with breakthrough technologies need to be embedded in patient care settings. And that's how we've developed a patient-first approach for research, really focused on improving the lives of folks with life-threatening diseases. And our main focus right now is cancer and sickle cell disease. One of the things that I think we have done at Christiana Care for a long period of time, but has been brought to light recently, is to emphasize equity and bring in the latest discoveries to diverse populations that don't normally have access to these. And we call that breakthrough technologies never seem to reach underserved populations. So the next slide makes me feel quite old. I wasn't going to put a slide on here, but I know John did, and with his wonderful picture. So I decided to give you a little bit of background on where I've come from. I have run a university research-based laboratory for about 30 years, and the main focus of my lab for about 20 of those years was in discovering and uncovering how proteins involved in the DNA repair and recombination work, and then sort of morphed into gene editing as the application side of DNA repair. And as I said, since 2015, I've been the director of the Gene Editing Institute, and I have faculty appointments at the University of Delaware and the Wistar Institute. Okay, next slide, please. This is evidence that I've actually been here a long time ago. And the reason I put this in here was that gene editing actually did exist before CRISPR, and that's the era of the Bible we call BC. Now, on this journal cover, which is Journal of Clinical Investigation, I'm listed with a bunch of other outstanding scientists. And I have to tell you, in about 1999, this was about the entire field. With the advent of zinc fingers, talons, and CRISPR, this probably makes up 1/100th of the current field, maybe even less. So, field has grown quite dramatically in 20 years, obviously, and the advent of CRISPR made it grow much faster. So, on the next slide, really sort of describes what we do in the Gene Editing Institute, and there are 3 missions: discovery research, education and outreach, and translational research. This actually has a great deal of importance for working with you guys on the education and outreach side, because CRISPR in a box was actually developed in the discovery research side and continues to be used to this day. In fact, this morning I was on a call with the group that is in discovery research, and they found some interesting new applications for the cell-free extract. And if you begin working in this area with this kit, you're going to have access to all of this, and that's what I'll be talking about a little bit later. But the three areas actually have an interesting piece together, where discovery research is focused primarily on uncovering the mechanism of CRISPR, the mechanism by which we understand how it works. The education and outreach piece, as I said, is really goes just beyond CRISPR in a box tool. It goes beyond a full experience. And then translational research side is actually our application side, where we are developing an FDA protocol for using CRISPR to treat squamous cell lung carcinoma. And we have a genome engineering team, which is happy to make cell lines for research institutes and universities around the world who seek to understand genes or how certain genes function. The partnerships that are listed on the next slide actually are some of the ones we're most proud of. Each section actually has a series of partners. Our discovery research is funded by National Institutes of Health, National Science Foundation, and a couple of private foundations, including the Lisa Dean Mosley Foundation. And this work actually drives a lot of the discovery pieces that we incorporate into our educational module. And as I've listed on that side there, and I won't go through it in great detail, Deltec is really at the top of the list. It's been a wonderful partner. We've been able to have great National Science Foundation support. And at the bottom is something called DETV, which you might think is an interesting partner in the education space. And I'll certainly have more to talk about their role and our upcoming equity and trust part of the CRISPR in a Box program. Next slide, please. So I just want to sort of talk a little bit about in 3 slides what the other parts of what we're doing, because I want to emphasize that in working on this particular aspect of the CRISPR in a Box kit, you're actually going to be working in a system that actually is still active in our laboratory. Part of it is helping us uncover a novel gene editing approach to lung cancer. Next slide. We also have, as I've mentioned, some very good partnerships with the Wistar Institute, where we actually make cell lines for people looking at drug discovery. And those people sit right next to the folks doing the educational development piece. So they're hearing and exchanging ideas and information, not only in how to make CRISPR work more efficiently, but the people in the education group are actually understanding what's the latest and greatest in the field. And we can transmit that very quickly into the education side. Next slide, please. And then we also have a national program where we do webinars talking about some of the best ways to use CRISPR. This is Pavel Bilk. He's the head of the Genome Modification and Engineering Group. And Indigo Johnson, a research assistant, has actually been did some wonderful work with us in generating cells. And so we give national webinars, fundamentally just talking about how CRISPR can be used in various applications. And the next slide, please. So here's what we really wanted to talk about. This all started with what we call a partnership in education. And it was basically created by 3 of the 4 people up there. I'm the guy in the suit, which is not, I probably have to change that picture since everyone else looks so casual. But the other 3 folks have really grounded this whole approach. And Kristen Pasarczyk is in the upper left. There's John. And then Natalia Rivera-Torres is our lead scientist. Dr. Torres is our lead scientist on the sickle cell program. And she'll circle back at the end to tell you how she's going to help in this whole new gene editing education program. Next slide, please. So I want to just spend a couple minutes kind of bringing everybody into play here and tell you how since we've developed this CRISPR in a box, and how do we think about gene editing? Well, we think about it as a genetic spell check where you can cut, paste, or change letters, genes, words, phrases, using genetic tools such as talons, such as zinc fingers, and of course our best tool, CRISPR-Cas9, Cas12a. And we can delete, insert, and replace segments of genes, and change gene function and change phenotype at a level and at a precision that has not been previously possible. And in some ways, having been in the field for 30 years, was probably not even dreamed to be possible. So a remarkable achievement in biotechnology, and obviously with the Nobel being awarded this year to Jennifer and Emmanuel, we couldn't be happier. It's a wonderful time to pull things together. Next slide, please. So I always put this slide up to kind of tell you where we've come in 20 years. Having been in the field and still, actually, we do a fair bit of work in DNA repair enzymes and trying to understand that enzymology. Excuse me. 20 years ago, we were on the left-hand side of this chart, and we were working with some pretty primitive tools. But as things evolved over the last 20 years, we're now at a very complex Swiss Army knife. Now, full disclosure, I have no idea if this is the true evolution of the Swiss Army knife, but I do know it represents the evolution of the gene editing toolkit. And that toolkit is, I think, represented on the next slide. I've been talking about zinc finger nucleases, talons, and CRISPR-Cas9, and some of you may wonder, why is it that we basically talk about CRISPR, CRISPR-Cas9, or CRISPR-Cas? This is a very informative slide, and I want to walk through this just for a little while to tell you how our research teams in our institute and in others around the world think about these specific tools. It's also kind of cool because you get to work on really cool sounding biomedical tools of zinc fingers, talons, and CRISPR. Designing, building, speed, and scalability are four fundamental aspects of using genetic tools. And you can see from this chart that CRISPR-Cas, which is really the only naturally occurring tool, actually has the most applicability. And as we've developed and as we have been meeting with the FDA, essentially scalability turns out to be probably the most important thing here. You may not have guessed that, but the ability to make enough of it to treat a human disease in the amounts that we need actually has to be readily possible. And so the CRISPR-Cas program has actually grown in such a way, and the work on that has exploded because of these four fundamental facts. Next slide, please. So CRISPR actually doesn't work as gene editing in bacteria. I think everybody knows that. It actually is a naturally occurring process known as adaptive immunity. And its job is basically to fight off viral infections. It uses a CRISPR-Cas complex to basically destroy these viral complexes. And there's a mnemonic behavior in the CRISPR-Cas genome organization so that when the same virus infects again, it attacks it even more quickly. But the Nobel Prize was actually was awarded to Jennifer and Emmanuel basically because they took a naturally occurring process, adaptive immunity, and converted it to gene editing. And as you probably all know, the enzymology in bacterial cells and mammalian cells is very different. And take it from someone who's been trying to figure out the mammalian pathways for about 30 years. So next slide, please. So here's the beast in itself. We think about it as being a two-component system, and there are variations of this that are so complex they actually make your hair hurt. There are a lot of graduate students spending their time in the bottoms of gold mines and old fields somewhere trying to find unique bacterial species that have variations of the CRISPR-Cas complexes fundamentally so they can avoid the patents owned by Berkeley and MIT. But in almost all the cases, the complexes consist of two components: a CRISPR guide RNA, a piece of RNA, and a nuclease in the form of those molecular scissors. And those scissors are transported to a specific site on the DNA, shown on the right-hand side of this as it spools out of the chromosome, to make a specific incision and a double-strand break. That's pretty much what CRISPR-Cas does. The beauty of it, of course, is the fact that we can design the CRISPR guide RNA synthetically now, even as a single RNA piece, where sometimes two pieces of RNA occur naturally, to direct where those molecular scissors work. Most of us who've been in molecular biology, even in some of our courses and our research, have spent our entire lives trying to knock out nucleases because they've destroyed our single-stranded DNA templates or our PCR primers, or even RNA if you had the misfortune of having to purify RNA in the old days. But they do serve a purpose, and essentially with the development of the gene editing protocol, CRISPR-Cas has been repurposed for basically doing this activity. This is not its normal function, but in mammalian cells, it's been democratized and pulled forward into that. Next slide, please. This is a diagram that a graduate student named Kevin Blau actually kind of pulled together a variety of different structures from around the world. And Kevin is a tremendous bioinformatist. And Kevin is going to, I'm going to show you how Kevin is contributing to the CRISPR in a box, so what we call gene editing 360 program, in a couple minutes. So, and essentially, those of us who are old enough kind of see the blue blob as a rudimentary Pac-Man. And the DNA binding groove is a remarkable feature of evolution. The DNA target binds in that site, and there are two shark's tooth indicated by the cleavage domains of the CRISPR that actually cleave down on the DNA once the specific guide RNA or the CRISPR RNA has positioned itself on the DNA. This is an important fact that I'm going to talk about right at the end of my section, because I want you to realize that the binding of the RNA, the CRISPR RNA, and the DNA target is essential. And we and other labs have shown that if you begin to disrupt that, if there are mismatches on the target, you can create some significant problems with the specificity and the efficiency of CRISPR-Cas activity. Next slide, please. So I've said that CRISPR-Cas gets a lot of billing because we can complete and cleave it specifically, but it's really the mammalian cell that does all the work. The enzymology baked into mammalian and human cells and other eukaryotic cells actually carries out either non-homologous end joining, where deletions through resection are created, and that's what we call genetic knockout, or homology-directed repair, which basically works to insert different pieces of DNA. And this is something that also requires a DNA template. It's not important to look at the circuitry that's displayed on this slide, but really just to tell you that 99% of the activity in gene editing is carried out by the mammalian cell. What the great advantage is that the CRISPR-Cas system allows us to tell those enzymes, those DNA repair enzymes found, in our case, obviously in mammalian cells, where to work. And so fundamentally, the combination of CRISPR-Cas and mammalian cells and DNA donor templates are essential for developing what we would call true gene editing. Next slide, please. So today we're announcing the evolution and the availability very soon of something we call Gene Editing 360. This is a comprehensive educational experience in gene editing, and the rest of my talk I'm going to tell you a little bit about the various aspects of this, and then John's going to come on a little later and go in specifically directly into CRISPR in a box and how that works. Next slide, please. Brett Sansbury entered my lab as a graduate student who has a degree in chemistry and was one of the few students who really wanted to work on mechanism. Almost all the students I see in postdocs and residents want to cure sickle cell disease or want to do cystic fibrosis. But Brett, because of her analytical skills, actually wanted to work on mechanism. And so together we sort of concocted an idea that we should try to do CRISPR gene editing in a mechanistic way. And we did this in a cell-free extract. And the reason we approached that, and the reason I gave this idea to Brett, was essentially because DNA repair, DNA recombination, DNA replication systems were pretty well described because of the use of cell-free extracts. And so Brett not only did this, but she created an absolutely brand new paradigm in the CRISPR gene editing field. And I think the next slide is sort of tells you the scope of what she's accomplished. Her work has been published and republished and cited throughout the scientific community over the last 4 years. And we were just able to persuade her to stay and lead the discovery group. Brett Sansbury works in the cell-free extract system, which is the CRISPR in a box system. And here are her publications. And at one of the aspects of our new gene editing, you're going to be able to have access to Brett for your students to talk to her about what she's working on and what she's driving her team forward. Next slide, please. Brett's work was considered to be one of the top 5 most downloaded articles in the premier journal in the field, in the CRISPR journal. And we felt this was very important, that if we developed an educational component, whether it's an experience or a kit or something, it ought to reflect what people are actually doing in the field. And that might be the best way to teach students interested in this field to actually use something that folks are really actively using. Next slide, please. So here is the simplified version of what we're doing. As I said, John's going to go into great detail of this, and this is the kit that's available from Rocket Immunochemicals. But essentially, the Cas12a protein and the CRISPR RNA are assembled into an RNP particle. They're put with plasmid DNA, and you can then look and teach how mismatched bases, if you get your CRISPR RNA sized and changed and sequence changed, how that is really essential for DNA cleavage. We then add a mammalian cell-free extract, and we have made cell-free extracts from just about every cell line, and they work interestingly to different degrees. And even among people, interestingly, they work to different degrees. And that's a very important piece to understand when you're teaching about gene editing and demonstrating in the laboratory. The gene editing activity among different patients and people is very, very different. So people shouldn't go away just assuming that it works the same way. And again, I'll mention this briefly toward the end. Essentially, we recover the plasma. We do a simple bacterial transformation, and we do bacterial read, a standard circle. Next slide, please. We've also developed a keen sort of curriculum for all of you. There are lots of curricula out there. We have a 2-week curriculum, which is illustrated here, and it kind of talks about some of the features that you could do. Plasmid cleavage with Cas9 or 12A, and they do work slightly differently. You can also use the cell-free extract to look at the activities of non-homologous end joining in HDR. Bacteria don't have non-homologous end joining, and so it's one of the unfortunate features of working in mammalian cells. These are competing pathways in the gene editing reaction, and people have to understand, as you'll see later, and something that Brett Sansbury discovered, that there is a diversity of outcomes with gene editing. You can also compare unedited to edited colonies and confirm genotype analysis, etc. Next slide, please. We've also developed a 15-week curriculum, which is about ready to be published in a Nature Protocols book that is just coming out, we think, in a month or so. In fact, ironically, we just received word from the Springer that publishes all the Nature journals that today they have just started the production. And the work was done by Kristen Pisarcik, who is a graduate of Delaware Technical Community College, who acts as our science educator. And you will have access to this paper for free. So this describes all of the things you see on the left-hand side and the table, as well as the specific types of activities that CRISPR in a box can do. Next slide, please. I'm just putting this up here. This is an actual piece of data that came from a work that Brett Sansbury and Amanda Hughes did in the lab. And I think this is quite informative and something that we can actually help in sort of teaching and expanding this. When you use gene editing in mammalian cells and in human cells, obviously, there is not always the exact outcome you hope for. In fact, most of the outcomes are quite diverse. And if you look on the right-hand side there, you'll see a series of green bars. And when you see green bars, and we've helped you see them by this huge red star, that's where the insertion of 8 bases or an HDR reaction has occurred perfectly. Now, in the perfect world, we would think that was it. but you can see that the world is not perfect in gene editing, and this is something that's extremely important to understand. And that's why where you see red and yellow and gray and light green, those are indications of mutagenesis, and this is the activity that occurs around the target site. So with CRISPR in a box and this whole sort of approach, you can actually look at these kinds of things in a variety of different ways. And the indication of sequences on the end, on the left-hand side, are actually available from us too. So we'll talk a little bit more about how to analyze those sequences in a minute. But essentially, this is a very important point that is significant in teaching about gene editing in general. While CRISPR is revolutionary, it still doesn't do things only in a precise fashion. Next slide, please. So we noticed this, Brett and Amanda had done some of this work, and we sort of noticed this, and we thought we would be able to pull together an interesting and important component of analyzing. But we did some of the bioinformatics and software programs that were out there, and they were pretty inefficient at analyzing Sanger sequencing, which is still used primarily when you're looking at very specific targets. And so our laboratory, led by Kevin Blau and a high school student known as Rohan Kanchana, actually developed something known as Decoder, which stands for Deconvolution of Complex DNA Repair. Now, Decoder is a software program that will tease apart complex DNA sequences that are represented by the milieu of outcomes you saw on the previous slide. And if you go to the next slide, there'll be a wonderful picture of Rohan and Kevin standing there. And these guys have not only perfected this in a way, but this is actually going to be licensed by a couple of major companies to do their own screening on their private databases. We're going to make this program available to you at no cost. I'll provide you with a link at the end. And if you don't want to do DNA sequencing from CRISPR in a box, we will give you files of thousands of experiments that Brett's group has done, so you can demonstrate decoder activity in a bioinformatics extension of the gene editing experience. Next slide, please. Now, one thing that I've learned as I've been in this field for many years is that gene editing isn't just about the science. We have a responsibility as scientists, whether as instructors or as bench scientists or as administrators, to make sure that breakthrough technologies not only reach underserved populations, and when will all patients have equal access to them, but also that they are used responsibly. Jonathan Marone is an ethicist and a colleague and a friend of mine from Harvard. And Jonathan and I have been writing about this for some time, and Scientific American asked us to write an op-ed about this particular, in this particular space. Next slide, please. So we've begun to think about it not only in terms of reaching underserved populations, but wrapping the ideas around ethics. Not only do we have a responsibility to do good science and report the truth, like the imprecision of gene editing at times, we also need to address these ethical questions. Now, this little fella here getting blocks together, could we engineer him to be a sprinter, to have a perfect pitch, to have 20/20 vision, to increase IQ? Well, for some men, we might want to increase the lack of baldness, so that could be a positive thing. There might be some votes for that. And while most scientists in the world are now fairly responsible about staying away from these kind of traits, and they're very complex, so the chance of doing this is low, these are the challenges that your students that we think and we even think are going to be facing. And these are the kind of questions that the general public wants to know about. Next slide, please. This is another piece of information that's essential for understanding CRISPR. And we wrestle with this all the time. We have a major, heavily funded program to develop sickle cell disease, develop CRISPR as a therapy for sickle cell. And this is what we encountered. As you know from the mechanism of how CRISPR works, in fact, CRISPR actually has to know and define the perfect sequence so that you can design your CRISPR RNA to bind to that specific sequence. In many cases, the public genomic databases don't contain contributions from many ethnicities. In fact, about 70%, sometimes even as high as 85%, of the deposits in these genetic bases are from European Caucasians. So if we're going to develop a genetic tool as a broad-based universal tool, it could be, unless we recognize and address this problem and talk about this problem and encourage people from minorities and underserved populations to engage more in public database submission, that CRISPR in itself will only be effective against a certain group of people, and none of us want that. Next slide. So we're in the process with Delaware DETV of producing a 6-session video module that'll be focused on what we call gene editing for sickle cell disease. We consider this to be the Gene Editing Institute's teachable moment. The first session will be sickle cell disease, led by a physician from Christiana Care who is treating sickle patients and likely to have interviews with sickle cell patients. Natalia Rivera Torres, who leads our gene editing and sickle cell disease, will then talk about CRISPR-directed gene editing. We'll then go through a CRISPR in a box demonstration, sort of like a cooking show. We're then going to talk about gaining trust in breakthrough technologies, and that'll be given by a wonderful woman who works in health disparities at Christiana Care, who is dealing to break through these kinds of things. And then we'll talk about ethics and regulation. And then at the end, our last session will involve members of the GEI team, scientists of every ethnicity, every gender, sitting around telling their stories of how they got to be in the Gene Editing Institute and what attracted them to gene editing, sort of talking about their journey to success with lots of failures along the way. Now, I've been told anecdotally that they're also encouraging them to talk about the frustrations they have, but I'm sure there are no frustrations in the Gene Editing Institute. But knowing my team, they'll be honest and transparent. This 6-session program will complement CRISPR in a box quite nicely and will be available for absolutely no cost on the GEI website very soon. They're in production right now with DETV, which is an African-American owned public access cable network that has been wonderful to work with and is able to communicate with that population quite nicely. Next slide, please. So we're getting some attention, and I think it's good. I'm not a big fan of public relations and PR, but one of the things that we're trying to promote as the Gene Editing Institute, circling around CRISPR in a box, is CRISPR for diversity and humanity's sake. We don't consider this to be an important piece of just instruction. We want it to be part of the general conversation about improving access. And you can do it by starting with a breakthrough technology that is revolutionizing how we think about genetic diseases. Next slide, please. So here it is. CRISPR in a box is at the center of the world for us, and we're improving it constantly. We'll have a new version of it where we'll include a lot of the accessory buffers and other things that have been left out initially. This rose from our wonderful partnership with Delaware Technical Community College, and we're going to run in parallel with their workshops. I believe they have one next month, where instruction with this particular reaction will go. And that's where CRISPR in a box kind of arose. There was demand for the reagents that they were teaching in there. And then we suddenly realized that it had some popularity. So we're going to expand, Rockland's going to expand the offerings so that you'll be able to purchase this along with all the accessory components you need to make it work. But we don't want to stop there. You'll have access to this for everything on this slide is on the outside is free. Gene editing for sickle cell disease, a breakthrough technology and a matter of trust. The 6-session educational group will be available to you. You'll have free access to Decoder, which can be used to untangle this DNA sequences that are generated by the mammalian gene editing system, the CRISPR in a box. If you don't want to get to the sequencing piece, we will send you files of DNA sequences that we've encountered using CRISPR in a box or using the cell-free extract. You'll have input with CRISPR in a box 2.0, 3.0, 4.0. What we've talked about and what people call HDR is only the beginning of the story. Point mutation repair, that's something for curing sickle cell disease. or inserting 3 bases for cystic fibrosis correction. Those are the kind of things that are coming, and it's all made possible because of the flexibility we built into CRISPR in a box. That paper describes all of it and the history, and that, of course, is available. But something that I'm really proud that we're going to be able to offer is interactive research updates on a monthly basis. These will be videos that we will produce, led by Brett Sansbury, the originator of this reaction, and her team, which is much larger than this. But you'll be able to hear from Brett and from Amanda and from Kevin, probably from Natalia and Sharon and Sambi, and folks who work in this area to talk about what they're using the CRISPR in a box reaction for. So folks who are tuning into this will be able to see how not only is this an instructional piece of educational inside inside your laboratory, this is actually being used on the front line. And I might even stop by, although I'm sure Brett won't allow me to say too much. So we're very much looking forward to this kind of interactive mode. And who knows, if this starts to go well, we might even do some live streaming so we can engage with conversations with these guys from students all over the country who are using this and have some questions about how they can improve. And we just love to hear the modifications you guys are thinking about in using this kind of a reaction. Next slide, please. And to do all of this, we've had to turn to one of our top people, leaders in the Gene Editing Institute, and this is Siobhan Hawthorne. Now, I was going to put her home phone number on there and her cell number, but I think it's sort of dangerous to do it. Siobhan has done a marvelous job pulling Gene Editing 360 together, and I couldn't be more proud of how she's done this. These are her contact information. If you'd like information on any of this, how do you access this? How can we work together? Her contact information is here, so please reach out to her. I'm just a phone call away with her, and I talk to Siobhan probably 2 or 3 times a day. So she's heading up the entire Gene Editing 360 team, so please reach out to her if you're interested in this. Next slide, please. And I thought I'd show you the team that has worked on this for a long time. Now, full disclosure, this photo was taken in August of 2019. I know that Dr. Nick Petrelli, who was a tremendous advocate for us in the Cancer Center, would have a heart attack if he didn't see everyone wearing a mask. But this was taken a time ago. The first thing you'll notice about this group is that it's an extremely young group of people. And you can tell the 2 people in the coats. They're Dr. Byung-Chun Yu, who is the associate director and essentially leads our lung cancer program through the FDA, and then me huddling in the back there. The rest of the folks you've kind of seen. And I also want to talk just very briefly about 3 people who are not pictured. Deb Vernon Snyder is the finance person in the Cancer Center. And for the first couple of years as we went forward, Deb was a true loyal component and an advocate for us. And without her establishing our financial situation, we wouldn't have been able to progress this far. And obviously, great thanks to Nick Petrelli, the director of the Cancer Center, who believed in early on that it was very important to put this remarkable gene editing institute inside a cancer center. And the rest of these folks have been a wonderful team. Anecdotally, somebody told me something very funny that I'd looked at this picture for probably 2 years, 2.5 years. And you can see that the gene editing group, the women are in the center and the men are on the outside. So anyway, so I'd like to thank all of you for listening to me. And what I'm going to do is to now turn it over to my colleague, John McDowell, who has probably been as good a partner in advancing this as I could imagine. John and I have known each other for a long time, and it's just been a real pleasure to have complementary skill sets working together on such an important endeavor. John?
Great. Well, thanks, Eric, for that introduction. Tough to follow on after the specifics, but what I would like to talk about is kind of the role that and the experiences that we have had in introducing this in vitro gene editing reaction that is the basis of the CRISPR in a box kit with our students in the community college level, as well as experiences that we've had with faculty members at community colleges throughout the country, and we're looking to expand that as well. So my quick introduction slide is shown here. I am an instructor at Delaware Technical Community College, which is the only community college in the state of Delaware. So we're small, but we have 4 campuses spread throughout the state. I'm an instructor in the Department of Biology and Chemistry. We have 2 AAS degrees in the program related to biology. We have a biotechnician training program, as well as a biological sciences program, which serves as our articulated degree program. I've been with the college since 2012. In the middle, I've taught virtually all the biology courses that we have in the program, while we are now able to bring CRISPR curriculum to at least the classrooms for all the courses that we have throughout the program. It's really our capstone level genetics and molecular biology, where students get the hands-on laboratory training, and in the genetics course specifically, where we have students that go through the process of the in vitro gene editing reaction. Eric's talked about some of the other grants that we have feeding into this, and mentioned that a lot of the activities that we have related to gene editing curriculum development at the community college level has come from support of the NSF through 2 ATE grants, one which we're in our current no-cost extension year, as well as a new one that was awarded just last summer. And so we're very excited and proud to be able to do the work. And looking at the names of people that are coming through, we have a couple of past participants of our workshops that are here too. So thank you for being here. You've been a big help as we move forward with all of this. If we could get the next slide. I won't spend a lot of time going through this. Eric described the entire process, but I did think it's important to highlight and kind of just refresh everybody's memory about kind of this flow. I think as anybody who has had a background and looked into CRISPR, as Eric talked about, it is the simplicity and the straightforward mechanisms related to gene editing, which makes it such a powerful tool. That's true in all of its different clinical and scientific implementations, as well as from a curriculum standpoint. So I think that CRISPR activities are one of the areas that we get the greatest level of student engagement and excitement over. The fact that we had the Nobel awarded for describing how this is being moved forward to gene editing is something that in the classes that I taught increased the level of excitement. But because it touches upon such foundational pieces of biology, we're able to implement it into multiple different courses and really touch upon many of the different themes and pieces of a curriculum that we need to cover. And so we're able to do that pretty straightforward through lab activity trainings like the in vitro gene editing system. I'm going to talk about the details a little bit of how the entire in vitro gene editing system works coming up, as well as some educational components and some thought pieces and some experiences we've had with students and the pieces that they really connect with. But as mentioned before, this is kind of the general overview. So hopefully that's been a good refresher for everyone. Okay, next slide, please. One, and there are many benefits of using the in vitro system, one of which is if labs are set up to do kind of general microbiology type activities that would be part of any type of micro course, if you have a nursing microbiology program, if you have biology programs, et cetera, the equipment required to do it is pretty standard. And so we've got just a picture and display of some of those things that are needed. All of this is taking place in a tube as an in vitro reaction until it comes time to get the readout. All of that is done in E. coli following a transformation. So if you have the materials to do a bacterial transformation, you have the tools necessary for doing the in vitro reaction as well. Next slide, please. So about that reaction, these are the couple of steps, and I think it demonstrates very well the fact that this lab can be done within a typical laboratory time period for courses that you may be teaching. So students going through this process start out by making their CRISPR-Cas RNP complex. So reinforcing the idea of that you have to have the walking gold man with the scissors from the picture that Eric described before. You need to have the two components of the CRISPR guide RNA complexing with a Cas nuclease. For the CRISPR in the box, we utilize the Cas12a nuclease. A dilution is made from the provided stock. Dilution is made of the specific CRISPR guide RNA. Those are added together to form the complex, which is done at room temperature in 15 minutes. And that's probably on the long side because there's an incredibly high affinity for the interaction between that CRISPR guide RNA with the Cas nuclease. So at the bottom of these next upcoming slides, you can see a couple of teachable moments, if you want to think about them that way, items that reinforce or can be used to reinforce what we're including as part of the coursework. So although the kit comes with a specific CRISPR guide RNA, good idea to go through how that was developed, what pieces need to be included sequence-wise to develop one of these CRISPR RNAs. It can be expanded into coursework where they use computer tools that are available to do that type of analysis, et cetera. Secondly, picking an appropriate nuclease. As Eric described, his team utilizes multiple different Cas nucleases. Here with the kit, we've got Cas12a. Cas9 has received a lot of attention and is used frequently as well. So this helps to provide a comparison between the different types of nucleases and an opportunity to talk to students about why certain nucleases may be chosen at some times versus other times. Finally, you're putting together RNA and a protein to produce an RNP. RNPs, this specific one is meant to go target and cut DNA at specific sequences, but RNPs are involved in anything from ribosomes, combination of RNA and proteins together with function, as well as RNA processing enzymes like SNRPs, et cetera. So depending on what components you have already in the curricula of courses you're teaching, this is just an offshoot of tools and proteins that have been discovered and talked about for a while and maybe part of the classes that you're teaching. Okay, if I could have the next slide, please. Now that you have the gold man set up with the scissors, it's time to go out and do the cutting. So in the next step, we've got the cleavage reaction. So following that 15-minute incubation, a tube is set up with the appropriate buffer, RNP complex, and then the target DNA, which will be cut by the RNP product. We, as part of this kit, are cutting the LAC-Z gene found on a plasmid. So that's indicated here. This is a 15-minute reaction. So the RNP finds its target, digests both strands of DNA at a specific location within the LAC-Z gene. You know, you've got the cutting. If you could go to the next slide, please. Another benefit of using this in vitro reaction is we have the ability to take snapshot views of the progress in the reaction. So as opposed to other methodologies where an RNP may be introduced into a cell or DNA may be introduced into a cell, which would then be transcribed to make CRISPR RNA or transcribed and translated to make the Cas nuclease, all of that activity takes place in the cell, which is sort of a black box until you get ready to see the phenotypic readout or the DNA sequence following the reaction. With the in vitro reaction, following the cleavage of the plasmid, we can actually take a small amount of that sample, have an agarose gel run, and compare the products of that versus the uncut plasmid to confirm that our RNP has in fact cleaved the plasmid sufficiently that we've got a linear form of the plasmid. And again, depending on the necessities of your courses, if you're already talking about circular plasmids, different types of conformations, how they look supercoiled versus linear, running agarose gels as part of your labs, these are all things that can be done as part of this process, both to look at this particular experiment as well as cover those basic fundamentals of the courses. Next slide, please. All right, so we've cut our DNA. And the goal is gene editing. So we're hoping to actually modify the DNA of the LAC-Z gene, not just have it cut. So in the next steps, we take our digested plasmid, cut DNA. As part of the kit, there is a single-stranded oligonucleotide, which serves as the template for homology-directed repair. We have the in vitro reaction buffer. And as Eric talked about, it's essential to have the enzymes to actually do the DNA repair at that portion. As part of that, cell-free extract collected and purified out of mammalian cells are included in that reaction. So in that cell-free extract are all the enzymes which will drive the repair of the cut DNA. If it utilizes the single-strand oligonucleotide, will drive it towards homology-directed repair, although there's the possibility of having non-homologous end joining occur as well, because there is that competition that Eric described going on inside the cell. So for students, a couple of the teaching moments are described below. Talk about what cell-free extract is, which enzymes are essential for doing these types of repairs activities, what would be found within each of the different cell types. Eric talked about the fact that different mammalian cells have different activities and how that reflects, could be reflected in possible clinical applications of CRISPR going forward. Discuss the important role of these oligonucleotides for directing homology-directed repair. The fact that you've got non-homologous end joining is another possibility. This particular oligonucleotide has an insertion, a sequence of NOT1. So if repair takes place, a unique NOT1 sequence is incorporated into the LAC-Z gene. This leads to the disruption of the gene, which is important for the phenotypic readout following transformation. It's also an important key because with that unique NOT1 site, we can use purified plasmid following this reaction and have students go through and do a restriction digest to determine whether that site has been added or not. Okay, and then we also have repair outcomes that are available as well. Okay, next slide, please. So once we've finished through that example, that last step of having the directed repair, recircularizing the plasmid, next step is just a transformation into E. coli to complement the LAC-Z gene. Plate the bacteria on XGAL and IPTG containing plates, let them grow overnight. And this particular slide shows representative data out of the in vitro reaction being run. So you can see that there's a mixture of blue and white colonies. White colonies are indicative of having an altered LAC-Z sequence following gene editing. So those would be positive. The blue ones, indicative of not having a disruption of the LAC-Z gene, so not the type of gene editing activity that we were looking for. But as Eric talked about, when you look at the DNA sequence from all of those, you actually see a broader spectrum than just working or not. So one level of going through this particular experimental flow is to look at the phenotype readout. You've got blue and white colonies. From being in class, this is an incredibly powerful visual for students to see when they realize that the process of CRISPR-Cas gene editing is not giving 100% perfectly edited results. You're not getting the intended result 100% of the time. In fact, there's some variability that goes on. And so when discussions come up with where we are at the state of the field, how they would recommend these for themselves or family members and think about it, the results speak volumes to the types of questions about where we are right now and what we can see. So if you're doing the experiment, this is one potential stopping point. You can go further, and I will discuss that with the next slide. So following the growth of the E. coli, the plasmids from individual colonies can be purified. Because of the unique NOT1 site that's included in the template DNA for homology-directed repair. Those plasmids can be digested with the Notl enzyme. And if editing has occurred and the entire sequence has been included into the edited DNA, you can see a digested plasmid, linearized, indicated by the higher bands in lanes 1, 6, and 10. In cells or in plasmids where there was not incorporation of the desired insert DNA, you would not have digested plasmids. The supercoiled plasmids would run lower, and those are indicated in the other lanes 2 through 5 and 7 through 9 on this particular slide. So again, many courses have requirements for teaching restriction enzyme, how they operate, and including those in lab settings. Students may be reinforcing or maybe reintroduced to those particular lab techniques following this step of all the process. Okay, next slide, please.
John, you did have a couple questions come in. It looks like Kristen may be addressing them, but I just wanted to draw your attention to that in the chat.
Okay, and I do see that. Thank you for pointing that out. That both Brett and Kristen have come in. So the plasmid question came up. Does it have a selectable marker? It does have, as pointed out, the plasmids that are used here have a kanamycin resistance gene as a selectable marker on there. So the media would contain the appropriate kanamycin antibiotic to select for incorporation of the plasmid. The blue-white is just an indication of whether there was editing specifically at the LAC-C locus. That was great. So thank you, Heather, and thank you for Kristen and Brett for responding back through the chat as well. Okay, next slide, please. Eric mentioned that the gene editing activities can really be incorporated in multiple formats between different classes, depending on the needs and the flexibility that instructors have in their courses. It can be done as a 2-week process. Most of these focused around the CRISPR in a box activities. The first week going through the first lab procedures to get bacterial colonies. The second week doing a plasma purification with a Notl restriction digest. In terms of lab activities, this is primarily how students in my courses experience the lab training related to this in vitro reaction. Next slide, please. It can be expanded in multiple different ways. This slide just shows some of the different kind of branching points that you have. So for individuals that are running a shorter length course over a couple of weeks, or that have the room to accommodate kind of half or a quarter of their semester, depending on how your courses are structured, there are multiple ways to engage with the kit activity and connect it with other materials which need to be covered in the course. And finally, on the next slide, as Eric mentioned, the work that Kristen and Brett have been doing in developing this larger curriculum is soon to be published. And in fact, you know, there are plans already kind of described here how these types of gene editing activities and related materials can actually be the basis of an entire course. Again, the analysis, understanding of DNA, activities of proteins, concepts related to information flow, et cetera, that are key components of all biology courses. All of those are touched upon by gene editing and especially CRISPR gene editing. So it is very easy to imagine and work towards developing those ties between these gene editing activities and the curriculum that would match what would be required in many different courses. Next slide, please. So just to kind of talk about what we've done at Del Tech, again, we have the benefit of a fantastic collaboration with Eric and all of the team at the Gene Editing Institute. We've been able to expose our students to CRISPR gene editing over the past several years. And you can see between 2017 and 2020, we've had 122 students from the college that have received this curriculum, and specifically the lab techniques associated with it. In addition to those labs, we have student-directed activities that feed into it, from design of oligos, design of targeting, bioethics discussions, literature presentations as part of the coursework. So their understanding of the current scientific literature is increased. As well as lab activities, the in vitro reaction, and we've also utilized yeast systems and mammalian cell tissue cultures. In addition to the student activities within courses, we have a robust undergraduate research program. So many of the students pick up some of the sequencing analysis activities from when this is done in coursework, to then have sequences which can be fed back to students in upcoming semesters for analysis. So we get kind of a cyclical loop with students that can get greater exposure to the techniques and understanding of gene editing beyond what we're already covering in courses. If I could have the next slide, please. Instead of me just describing what they get out of it, I will not read these, but maybe we could hold them up just for a second. These are responses that we've received from students that have had this training. As I said, it is a fantastic, engaging field, an area to be able to present and share with students. They agree with that, and in many cases feel greater self-efficacy, greater interest in the areas of science, and we've received feedback that that has influenced their future educational and career activities. This is also, of course, beyond one of the people that Eric was talking about, Kristen Pisarczyk, a graduate of our program that as a program student, undergraduate, was engaged in research activities with the Gene Editing Institute before going to work there. We've had the opportunity for several of our students since then to also get the opportunity to be in the Gene Editing Institute to gain experiences in research and working in that environment. Okay, next slide, please. As part of the ATE grant programs, we have held a variety of faculty professional development workshops. These are specifically focused at the community college level. Community colleges typically have lagged behind the ability to bring new cutting edge technologies to students. And so we have been running faculty professional development workshops to help with that dissemination in this particular area for faculty that want to gain the experience. And whether we've had 2-day workshops or 5-day workshops, all participants in the workshops that we've held have utilized the in vitro reaction, the CRISPR in a box reaction, as part of the hands-on lab activities, while also discussing curricula, implementation, and other instructional practices to have a greater impact in teaching. So to date, we've had 40 community college faculty that have come to these workshops. This is also the basis of gene editing activities at the Curry Lab Methods workshop held at Tulsa Community College in 2019. So an additional 12 faculty members, some of whom joined us this afternoon, were present and went through the lab experiments there. And again, we remain focused at meeting the needs of community college students using model systems that do not require extensive equipment in order to provide these experiences to students. One last point here, Eric mentioned, but we are planning some online webinar type workshops related to this, since in COVID times, we're not able to travel to my campus to be able to do this in person. We will be having, we plan to have one of those on February 12th and another one coming up in April. I'll give a plug now. Also, Heather, I think, is going to drop a link for a survey at the end for everybody. If you're interested in being on the mailing list for any of those upcoming workshops, please say that you would like to be contacted, and we'll be able to connect with you and give you an invitation to be able to join us. Okay, next slide, please. Okay, and the last thing to kind of bring up in here, again, with the ATE program, we've had these 2 different grants, which have been a great benefit to be able to run the workshops as well as to bring the curriculum to the students at Delaware Tech and share that throughout the country. And our program has been hugely successful and beneficial. And we were very honored that it was chosen as a project to be highlighted in last year's ATE Impact Books. And I think that was the final slide that I had with that setup. So I guess if we've got some questions, there's time to go through that, or
Yeah, we would now invite any of our participants to unmute, ask any questions you may have about the presentation. I'm sure that, John and Eric would also be more than happy to go into detail about any kind of classroom management or kind of the more finite details of how the kit works. So please feel free to join in and ask any questions.
I have a question. I don't know if I see myself anywhere. Can you all hear me?
Yes, we can hear you.
Okay, good. I wasn't sure if I clicked it right. I was looking at some of your results that you had left at 4 degrees Celsius and some that were at room temperature. And I noticed you still seem to have pretty accurate results. Would you recommend it at 37 degrees and then taken out after a day? Or if I presently don't know exactly when all my students are in the room, so I can have them for 2 days in a row, but sometimes some won't come that second day. So I would then have to take pictures of their data. Is there a period of time where it seems like contamination is too bad, or a better temperature to incubate once the, transformation part is done?
Well,
I can actually jump in and answer this.
I was
This
going
is
to say, Kevin
Kevin
or
Blau.
Kristen, please jump in.
You know what I mean?
There are easy answers here. I'm not sure the silence was weird.
Yeah. So the current, these plasmids contain a kanamycin resistance gene, so they are actually relatively stable with no satellite colony formation. At the 37 degrees, we've seen upwards of 24 hours with no satellite colony formation or anything like that. If worst case scenario, if you need to leave it out for multiple days if students can't be in, we've also left it out at room temperature in addition to the 4 degree, and it has also been fine without the formation of any sort of like contaminating colonies or any satellite colonies as well.
Okay. So it's pretty stable for a few days if it takes them a few days to get back into the room to come in.
Certainly. And I could probably add on to that too. If I'm not sure if Kristen's here anymore. She was the one, Kristen Pacharczyk, she's the one who actually did that series of experiments that showed the different temperatures. But even if, let's say you just leave it in for 1 day at 37 and you want to take it out and store it, we've kept our plates in 4 degrees, just putting them in there they can go weeks in there perfectly fine without any problems because of the antibiotic that's in the plates. Yeah, and even we see sometimes that if they're left in the fridge for a day or so, the blue pops even more. So
You get
it looks,
color, just like with
yeah, you'll get, yeah. So that's always an option too.
Okay, thank you. I was thinking about implementing it this year, and just wanted to work out some kinks with our new schedules of students in the classroom. So it's you know, so I'll do some part virtual and then some part in the room, and then maybe send them some results that they can analyze and such.
Yeah, we'll also, Danya, this is Eric. It's great to meet you. Finally, I've heard so many wonderful things about you. So we're also, as I mentioned, we're about ready to sort of put together this this package that kind of surrounds sickle cell anemia. And I know John has videos of Kristen actually performing this reaction. So those things we certainly will make completely available to you for free. So if you have to sort of piece things together where you want to run, you know, Kristen through an hour of her kind of going through the reaction and then teach it, so that will all be made. That's sort of what we're expanding onto the central piece of the CRISPR in a box thing has been what John has gone forward and taught. And our stuff now, because of our commitment to being more involved in gene editing rather than just a laboratory instruction or tool, those are all supplementary things, but they should be teaching helps and aids to help in the process. So those are all available for free. So those kind of things will be up on our website very soon. And as I said, we've partnered with DETV to sort of focus a little bit more on the implications of gene editing. And each one of those individual sessions that I talked about, those 6 sessions, are individual and they're self-contained. They are linked by a common theme, but you don't have to take all of them. You can take one at a time and say, you know, this time I need this piece and things like that. And then later I might want to talk to my students about, you know, how do you develop trust with a
I
breakthrough
think I can
technology?
incorporate it at different times.
Right, exactly. Yeah, that's what we're hoping for. They're sort of linked in that way. But we actually feel it's an interesting time, moment in time for us. You know, we've had some contributions to developing CRISPR, and we feel personally responsible, I think, for its proper use. And the way to I think it's going to be very important that those of us who are working on this at a very fast, intense pace also look to the side for the humanity side of this, and not just sort of try to accomplish the goal. So that's why we have done this. Our, you know, working with John, I mean, Deltec is a wonderful, you know, program themselves. We're able to supplement all of this because we see this and we deal with this. I you know, when I'm out speaking somewhere, when I used to be out speaking somewhere, when I'm sitting in my office speaking somewhere, it turns out that it's one of those questions I get all the time. You know, what are you doing to protect humanity against the you know, this you know, CRISPR actually,
Or get more humanity involved in
that is
understanding
exactly
it.
what we're doing. I couldn't have said that better. That is exactly what we're
That's my
doing.
goal.
Yeah, because it all wraps around that. And you know, during the Obama administration, John Brennan, who was the national security advisor, sort of indicated that CRISPR could be used as a weapon. And so educating folks not only about actually how to use it specifically, but also about the ramifications. We feel a responsibility to do that. And this is all arisen because John did these workshops and he's running those really well, but we can supplement, we can help high school, 2-year and 4-year colleges by getting integrated into the folks that are actually doing the work. Because it's a new kind of scientist coming along. You know, in my day, it was really about the results and things. And for the next, you know, pieces of time, the new generation of scientists in our group, Brett and Kevin, you know, was speaking there, and Kristen and Natalia, they look at things a little differently, and I'm leveraging the strength of their, you know, their perspectives on life nicely. So you get them, not me, which is good.
Looking forward to that.
Yeah.
Well, thank you for this. I appreciate it. I think it was
Sure.
very well laid out.
And I just want to jump in to clarify the website that you're talking about, these resources being posted on, is that the Gene Editing Institute website?
Yes, I'm sorry, Heather. Yes, it will be. We're slowly starting to load them up. Some of these things are still in production. It's absolutely brand new, but since I had an opportunity to talk to folks of your caliber, I thought, you'll start to see them come up, and we'll work through you or through John to let everybody know that everything is available. But we don't want to wait. We want to start putting things up as they come along. And I'm give a lot of lectures. But I don't run courses. It's a skill set I wish I had because it's very complicated. So how you're dealing with all of this, I know at each level and each school is probably very different. And so I think, as Danya said, interestingly, you can pull this one down. You might want to talk about trust here, or ethics there, or demonstration, and that's the kind of thing that we want to do. So we're sort of pulling that up in there. So yep.
Yeah, so I just put the link to the homepage of the Gene Editing Institute in the chat box. InnovATEBIO will definitely be keeping an eye on it and sending out an announcement when all of those resources
Are available
today are available. But in the meantime, if you want to bookmark that and check it frequently for updates. I mean, I just looked at it a little bit, and it's a fantastic resource, already. So that would be great. There have been a couple questions come through on chat. I think they've mostly been addressed. John has shared his email address if you're interested in the professional development workshop. There's also the opportunity to leave your information when you complete the survey on today's webinar, and those will be passed along, those email addresses, so that you get the direct information. I see that it was a question linked to Bio-Rad. That should have been updated to this, but if not, my apologies, and it is, the answers that come in from today will be forwarded to John for his sessions. And then we had a question about the cost of the kit. I believe that information is on the website.
Yep.
CRISPR in the Box website.
Yep. Yeah, that's at Rockland Immunochemicals. And as I mentioned, where, this was it was kind of developed for the workshops, and as we're expanding it now, we're going to add more things to it. But everything I talked about today is free of charge. The supplemental teaching materials will not, there'll be no charge for it. I just love volunteering people like Brett and Kevin to do videos once a month. So they'll I'm sure they'll be delighted. But we have 26 people, or we will have 26. So there'll be a wide variety of people there to chat about kind of what's going on, and that can be used. That kind of circles around this reaction, but also touches a couple of other areas too. So.
Yes, and I believe those resources, they're not going to be behind any type of firewall. So even if you haven't purchased the kit, you'll be able to go on and get those.
That's right.
Resources.
Yep. Yeah. Well, we can also do a crossing. DETV is also going to have the things that they do. They'll be fully available for that. We've gotten some very nice support for this whole initiative, and we think that just making this as a we're a nonprofit, so we're able to have some flexibility in what we do and dedication. So we can kind of be a little bit more flexible, perhaps, in sort of adding new teaching tools that work. And like I said, we're also very interested to have a conversation and information from you guys, that as your students use this, if there's some ideas that they have, we can certainly set up a moment or a time in which we can make some of our scientists available, maybe even with John there to talk about what they're working on directly. So you have, I think, as I mentioned, the conversations that we're having now are, this system has a lot to discover in it yet. So we are, and we're actively working on this. We actively use this system to define certain problems. So although it's an instructional tool, and we're delighted it works in a robust fashion, it also could be used to address unique questions. that are not just baked into it. It actually has some flexibility in there as well. So.
If there are any other questions, please feel free to unmute or write into the chat box. All right, well, I'm going to put the link for the survey into the chat one more time. Thank you all very much for coming today. Thank you to our presenters. It has been a great 3 weeks, and we are very pleased to have had such an excellent team come and present today, both from the kind of field research aspect that the Gene Editing Institute brings, and then Delaware Tech on the side of the two-year college students. So we're always available for any follow-up. You can feel free to reach out to me and I can make any connections, or you can contact John directly, as he's put his email into the chat box. So
Heather, I just want to
Yeah.
give a great shout out to two people who never get shouted out to, and that's Ashley Baker, who put this whole thing together and saves my butt all the time because she runs the slides, and she's our chief administrator in the GEI, and Danielle Zach, who kind of monitors all the financial stuff we do. So these two guys hate to be called out, so I'm going to pay for this on Monday for sure, but I did want to embarrass them publicly, and thanks to those guys for making me look good. So I appreciate it.
For sure. Thank you all for coming, and have a great weekend.